So today let us discuss about the clinico-pathologic correlations such as selected examples of cell injury and cell death. First let us see about hypoxia and ischemia in detail. So hypoxia, first let us see about ischemia. So ischemia is the most common cause of cell injury because of research from hypoxia, that is hypoxia is induced automatically in ischemia by reduced blood flow. When there is reduced blood flow due to mechanical artery obstruction or any other disease, the oxygen transport is decreased. So ischemia will automatically lead to hypoxia. But there is another condition in which though the blood flow is normal but oxygen level is less in the blood. So this condition is hypoxemia, or nothing but I can tell it as hypoxia. So in hypoxia actually, um, the though the let us discuss about ischemia first. In ischemia, it is different from hypoxia. How it is? The blood flow is maintained. So ischemia will compromise it. Let us discuss about ischemia first. So ischemia in ischemic tissues, both aerobic and anaerobic energy generation will also fail. Okay, because we need blood flow. Only then at least anaerobic energy generation will takes place, though oxygen is less in case of ischemia. Okay, so in ischemia the problem is that anaerobic energy generation will also fail because there's no blood. So without blood, how come glycolysis can occur? There are no metabolites, there is no glucose. So both aerobic metabolism and anaerobic energy generation is also exhausted, or else you can tell it as glycolysis is inhibited by the accumulation of its metabolite, and the final product is pyruvate. So accumulation of pyruvate will also lead to decreased ATP synthesis. Whereas in hypoxia, the blood flow is maintained. Unlike ischemia, hypoxia, the blood flow is maintained, and energy production is by anaerobic glycolysis can occur. So in hypoxia, anaerobic glycolysis still takes place in hypoxia. So in ischemia, it causes more rapid and severe cell. So that is why. So the question might come, which is dangerous, ischemia or hypoxia? The answer is ischemia. So in ischemia, both aerobic and anaerobic energy generation is depleted. So let us see some mechanism of ischemic cell injury. So in a certain sequence of events following hypoxia or ischemia, it reflects many biochemical alterations. For example, when oxygen level fails, the oxidative phosphorylation rates and ultimately ATP generation decreases. We can tell one example, like oxidative phosphorylation decreases and ATP decreases. And in another way, you can tell like the consequences of ATP depletion uh, results in reversible cell injury and later in cell death by necrosis. That is what we want to tell. Okay, so mammalian cells will develop protective response against hypoxia. How come is it is developed by the induction of a transcription factor that is called as hypoxia inducible factor. This will induce new blood vessel formation and it will enhance glycolysis. Okay, so HIF1 hypoxia inducible factor. Okay, next let us see about ischemia reperfusion injury. So in ischemia reperfusion injury, the blood flow, restoration blood flow to ischemia reperfusion, as the name indicates, this means there's a restoration of blood flow to the ischemic tissues. That's all. Simple terms. In simple terms, the tissue whose blood supply was lagging is again receiving the same normal blood supply. So that recovery of cells is possible if they are reversibly injured. But in case if they are irreversibly injury, then it will automatically lead to cell injury and cell death. Okay, so this is the ischemia reperfusion. So ischemia reperfusion is in clinical ways it is important. Why? Because it contributes to tissue damage, causes tissue damage during myocardial and cerebral infarction. So this ischemia reperfusion injury is important in contributing the tissue damage during MI. Okay. So let us see how does this injury occur. So usually we will think like reperfusion is distributed reperfusion, no cell injury occurs, but cell injury do takes place. We will see how it occurs. First is, mechanism is oxidative stress. So again suddenly the ischemic tissue is receiving the blood supply in ischemia reperfusion case. So in the ischemia reperfusion case there is a reoxygenation. So what happens is there is increased generation of reactive oxygen and nitrogen species. It is produced in reperfused tissue who's getting the blood. So due to incomplete reduction of oxygen in leukocytes, that is the main reason in leukocytes as well as in damaged endothelial cells and parenchymal cells. That is why the reactive oxygen and nitrogen species are produced, and this will cause cell injury. As we have seen earlier, these free radicals will cause cell injury. Next, this is intracellular calcium overload. So this is also we mentioned it. I explained it before. Intracellular and mitochondrial calcium overload starts during acute ischemia. But during reperfusion it is accelerated because calcium will influx resulting from cell membrane damage and ROS mediated injury to sarcoplasmic. So due to cell membrane damage or due to reactive oxygen species mediated injury to sarcoplasmic reticulum, the calcium ions will leak into the cytoplasm. So so this will cause cell injury. 817. So this will cause cell injury and also ATP depletion. So calcium overload mainly what is the consequences? Opening of mitochondrial permeability transition pore, and ATP leakage will occur due to which result in ATP depletion. Okay, that is important. Next, inflammation. The systemic injury is associated with inflammation because of danger signals which are released from dead cells. So there's a danger signal which is released from dead cells. That is cytokines. Okay, cytokines are secreted by macrophages and increased expression of adhesion molecules by hypoxic parenchymal endothelial cells. So it will attract. It's nothing but these danger signals and cytokines which are secreted by macrophages will cause accumulation of circulating neutrophils to reperfuse tissue, and this inflammation causing inflammation leading to additional injury. Next, so main thing is macrophages and cytokines and danger signals. All these three will contribute to inflammation by attracting the neutrophils, that is recruit circulating neutrophils to reperfuse tissue. Next, activation of complement system. It will contribute to the ischemic reperfusion injuries. For example, IgM antibodies have a probability to deposit in ischemic tissues. If IgM antibodies usually due to lack of blood flow, it may get deposited in ischemic tissues. But when blood flow is again continued, this IgM antibodies is bound to complement proteins, and IgM complementary proteins will bind to these IgM antibodies and are activated. So hence it will accelerate the cell injury and inflammation. Next is chemical or toxic injury. As you know that many drugs are metabolized in the liver, this organ is the major target of drug toxicity. So toxic liver injury is the we should remember toxic liver injury when a new drug is developed. New drugs should not cause toxic liver injury because liver is the site of detoxification of drugs. So if you give toxic drugs, then liver will get some kind of injury. First one is chemicals induce. Let's see how the chemicals induce cell injury. How it is cell injury. The first one is direct toxicity. So some chemicals will induce cells by combining directly with critical molecular components. In mercury chloride, we will see the example. In mercury chloride poisoning, just very fatal to life. Mercury binds to sulfhydryl group of cell membrane. That is what important. Mercury binds to Yes, which group? Sulfhydryl group of cell membrane proteins, and it will cause increased membrane permeability. This is harmful. If there is increased in membrane permeability, the polarization, the resting membrane potential in neurons is not maintained, and sometimes sodium ions will diffuse into the cell, and it will cause cell swelling, lysis, and so on. So, and also inhibition of ion transport 2 occurs. So, by these mechanism, the mercury will cause in mercury chloride poisoning, mercury will cause a great cellular injury. Okay, that is important. If you take cyanide poisoning, it's a cyanide poisons the mitochondrial cytochrome oxidase. So if it poisons the mitochondrial cytochrome oxidase, without that oxidative phosphorylation cannot take place. Cytochrome C oxidase is very important. It will inhibit oxidative phosphorylation. Next, conversion to toxic metabolites. So some toxic chemicals are usually inactive, but in a native form it must be converted to reactive toxic metabolites. Only then you can act on target molecules. So example is cytochrome P450 mixed function oxidase. This is an enzyme present in smooth endoplasmic reticulum of liver. The toxic metabolites causes membrane damage and cell injury by formation of free radicals and lipid peroxidation. Everything what we discussed is only given here, so you can refer to the previous video. In the next video we will be seeing about adaptations of cellular growth and differentiation. Thank you.